Jim. This is my first time holding the cube, so I hope everyone can hear me. Kind of weird, actually. I'm Newton Edelhofer. I'm a professor in this department. I also direct the biological station in Pellston. And it's my great honor to introduce our first early career scientist. Thank you for our mid-career scientist presentation, which was really a great start to this day. Fiona Soper has a bachelor's degree, a bachelor's of science with honors from the University of Queensland, 2006, and she earned a PhD at Cornell in 2016 with my friend and colleague Jed Sparks. She postdoced at the University of Montana for 2 years, and she's now a faculty associate at the University of Montana as well as a postdoctoral associate at Cornell. Fiona studies nitrogen fixation, and it's just, this is going to be very short because Fiona has all the information, but think about nitrogen fixation as a process and how vital it is to the living system. Before nitrogen-fixing organisms evolved, the Earth was probably taking about 10 million metric tons per year into the biological system because of lightning. Lightning could oxidize the biologically inert N2, create NO, NO2, NO3, and that would rain down into the Earth, and it could be reduced and fixed. Once biological nitrogen fixation evolved in multiple lineages, the Earth was pulling about 200 million metric tons a year out of the atmosphere, putting it into biological systems. Now, human beings are appropriating about as much nitrogen from the atmosphere as biological nitrogen fixation. About 60 million metric tons in agriculture, which we've appropriated biologically nitrogen-fixing species, and fertilizer. So Fiona, using stable isotopes to really understand controls on nitrogen fixation, downstream effects of nitrogen fixation, is doing really cool and important work. So thank you for coming. Thank you for accepting our invitation. Thank you for your presentation.
I'm glad to see, actually, you led into kind of my first point that I sort of share my enthusiasm for nitrogen fixation process. Because what I was going to say is that, I think every scientist gets to have a thing that they just illogically and kind of irrationally love. For some people, that's boa constrictors. Can you move your microphone a little?
Yep.
Oh, maybe I didn't turn it on. Oh, there we go.
There.
Is that better?
Much better.
Okay. And for some people, that's gravity or mitosis, and for me, that thing is nitrogen fixation. It's just something that, there we go, that I love. And because I would argue that after photosynthesis, as Newt pointed out, nitrogen fixation is probably the single most important process that supports life on Earth. And even though we're swimming in it, so it's 80% of every breath that we take, biological systems still don't have enough of it. So nitrogen still limits productivity in virtually every, as you can see here, terrestrial ecosystem, also marine, freshwater ecosystems sometimes. Biology doesn't have enough of it. And that's because taking nitrogen from the air, it occurs in these sort of triple bonded molecules, they're very sturdy, and converting it into nitrogen in a form that biology can use is really energetically expensive. And as Newt mentioned, there's only a small number of organisms that ever evolved to do this. And a lot of them form symbioses with plants. So things like soybeans and alfalfa and lupins. But in a lot of ecosystems, the most dominant nitrogen-fixing organisms are trees. And if you live around here in Michigan, that might be something like an alder tree. If you're in a tropical rainforest, many of the dominant flora there are nitrogen-fixing trees. If you think of the iconic African savanna, those kind of standout acacia trees are nitrogen fixers. A lot of the dominant flora of Australia are nitrogen-fixing trees. And although it's this really crucial process, it's not ubiquitous. So N fixation doesn't happen everywhere that it could happen all the time. And well, we know that because if it did, then we wouldn't continue to have nitrogen limitation. And so that means then that there must be controls on that process. And we're interested in these controls because they influence things like the trajectory of succession, for example, how much nitrogen is being fixed and when and where and why impacts the efficiency of agroforestry systems. It's a particularly big question for the outcome of how a lot of ecosystems are going to respond to elevated CO2. And so for that reason, we're really interested in measuring the process and then passing out what these controls are. You know, are they environmental? Does N fixation respond to the amount of nitrogen that's already in the environment? Does it respond to climatic factors, so water availability, temperature, seasonality? And then on the organismal level, I like that Jim set up a lot of the processes I'm going to talk about today. You know, does it respond to changes in plant demand during phenology or with plant development or plant size? And one place where questions about the nature of fixation are particularly interesting is in the case of woody encroachment. So Jim mentioned an example of where shrublands in the West are turning into grasslands. But as a matter of fact, that's kind of an anomaly. Across almost every grassland and savanna in the world, in the last 200 years, we have seen a massive increase in the abundance and the density of woody plants. So that process, it's called shrub encroachment or brush encroachment or woody encroachment. And it's hugely widespread. Globally, it's the second largest land cover change of the last century after deforestation. And a lot of the trees that catalyze those processes, especially in more arid areas, are nitrogen-fixing trees. That is the case, for example, this is Prosopis glandulosa. It's mesquite. You probably recognize it as a barbecue flavoring. And it's one of the most uncommon encroaches across the southern US. You can see here, this is some aerial photography from Southwest Texas, that in the course of the lifetime of some of the people in this room, some of these ecosystems have gone from open savannas to wall woody plants. And when Prosopis encroaches, it's establishing in nitrogen-poor grasslands, and it's still very much going on today. And so we have this corona sequence of plants that have established, you know, a few years ago up to 100 years ago. And as they encroach, they're really transforming the nitrogen cycling in that ecosystem. So here, for example, you can see that over the course of 150 years, the amount of available nitrogen beneath Prosopis canopies is doubling or tripling. They're changing greenhouse gas emissions, they're changing sort of the understory community, really transforming those ecosystems. And fixation is probably a big contributor to that sort of transformation process. And for that reason, we want to be able to understand and predict what is controlling nitrogen fixation during this process. Do we have more happening under specific environmental conditions, for instance? So those are interesting questions, but we have a problem. So fixation is also one of the hardest biogeochemical processes to measure. And that's especially true in trees. You know, they're large, they're long-lived, and very inconveniently, fixation happens in nodules underground. And so this is my former lab group in Northern Australia trying to trace one root from one nitrogen-fixing tree to find its nodules. And it becomes pretty obvious that that kind of process is not really sustainable. And any methods that involve accessing the roots are particularly difficult in arid ecosystems, you know, like, for example, the kind of ecosystems that Prosopis encroaches in, because the roots are deep and the soil is hard, and digging up, you know, your entire field site is usually not an option. And so, I'm sure everybody saw this coming, enter stable isotopes. So isotopes offer this really unique way of getting at the process of nitrogen fixation because of this one really convenient fact. So fixed nitrogen ultimately comes from the air. The air also happens to be the standard by which we define nitrogen isotopes. The air is 0 per mil. And unlike almost every other enzymatic process, which, you know, enzymes, they're trying to be energy efficient, and they will typically act on lighter isotopes first. And that process is called fractionation. So that if you have a pool of mixed heavy and light isotopes, once an enzyme is acting on it, it's preferentially taking out the light ones and leaving the heavy ones behind. But for reasons that we don't necessarily fully understand, fixation doesn't do this. It's unusual. And so we have a really good idea about what the signature of fixed nitrogen is. And it's really similar to the signature of the air. And so then if you have a nitrogen fixing plant and it has two potential sources of nitrogen, one of them that's really well constrained, the other one is soil nitrogen, and those numbers are very variable. We don't measure them all that often, but they tend to be much higher than fixation. And so if you look at the tissue of these plants, they're a mix of nitrogen that comes from these two different sources with an isotope value somewhere in the middle. And people have been using this, taking advantage of this fact. This is a method that's been around for several decades, and comparing a fixing plant and a non-fixing plant in the same ecosystem. And if you assume that they're sharing the same soil source, and then you compare the isotope value of their leaves, any difference between them should theoretically be proportional to some dilution from this fixed nitrogen source. Which I admit was a great idea, but it has a few problems and a few assumptions. So firstly, this only works on the scale of leaf lifetime. Leaves can live for months or years, and if you're interested in seasonal processes, for example, you're out of luck. You need to have another woody, ecologically similar reference species. And then you need to assume that these two different species process nitrogen in the same way, that they're taking up the same forms from the soil, that the way they're shuttling it around internally is the same, that the way the leaves are aging is the same, which seems, maybe dangerous. And then you have to assume that they're accessing the same homogeneous soil source. And if there's one thing anybody who works on ecosystems knows, it's that ecosystems are never, ever homogeneous. And so if you don't like the methods that are available to you, you've got a couple of choices. You can complain about it, which I did for a while, or your other alternative is to try something new. So I wanted to see if we could take these basic facts about fixation and apply them in a slightly different way. And so this method, I call it sort of fine scale nitrogen isotope sampling. You can think of it as being maybe more spatially and temporally explicit. And this method relies on the idea that instead of assuming that the soil source is constant, that you can measure the isotopic composition of plant available nitrogen in the rooting zone. And then instead of sampling leaves, it might be more useful to sample xylem sap. So xylem sap is it's containing recently acquired nitrogen that's on its way from the roots up to the leaves. And it has a turnover time that's more like, minutes, hours, rather than sort of months to years. And instead of comparing species that might be quite different to each other, the idea is that if you know what the soil baseline is here for a given plant, that you could look for divergences away from and then back towards that baseline in the xylem sap, and maybe interpret that as being indicative of changes in input from fixed nitrogen. And so that overcomes some of those kind of assumptions and limitations of that previous method. And I wanted to see if we could apply this method to this Prosopis encroachment system to learn a little bit about the controls on fixation. This is an arid system. Water and temperature vary a lot seasonally. Are there seasonal patterns? Which, also are interesting because short-term variation in water and temperature might let us learn something about longer-term variation that we expect with climate change, for example. Processes like drought in a water-limited ecosystem. And then as these trees grow and establish, and by the time they're 100 years old, growing in a very different ecosystem context, do we see changes in that dimension? The actual process of implementing this is mostly done at really unreasonable hours of the morning. You have to sample these things before dawn and do lots of sort of soil extractions and things, but that might be a good idea if it actually tells us something interesting about patterns of fixation. So, does fixation change as these trees establish and grow? As I mentioned, young trees, they establish into these sort of nitrogen depauperate grasslands. But then by the time they're 100 years old, they're large, they've got deep root systems, and they're growing in soil that now has a lot more nitrogen in it. And if you think that nitrogen fixation is energetically expensive, it might make sense not to fix as much when there's just more nitrogen already available in the environment. So we might expect nitrogen fixation to decrease over time. But then if we just look at the foliar, or in this case the xylem values of trees of different ages, not a lot going on, to be honest. Seems disappointing, but these trees are growing in really different soils, and so you can only interpret this if you know the baseline. And if we look then at that baseline, so how the soil nitrogen is changing over this period of time, it is showing a consistent upward trajectory. It's changing by 2 or 3 per mil. And if you remember that xylem sap is a mixture of this soil nitrogen and N from fixation, if fixation was a constant portion of the plant budget over time, we'd expect to see it tracking this line. But when you look at the difference between these two, you can see that over time, actually, they move further and further apart. And that's really only consistent with some extra input of isotopically light nitrogen, which looks like fixation, right? And that's interesting because it contradicts the mechanism that we thought was acting. So actually, it looks like maybe fixation increases over time. And there are a couple of suggestions for that, again, coming up to what Jim set up. Maybe there's differential survivorship. Maybe trees that are better at fixation have better survival rates, and so old trees tend to be higher fixers. Maybe water is a strong control. So, a small plant in a grassland might not have access to enough water to realize its fixation potential in a way that it does once its roots become much deeper. But either way, this more detailed sampling reveals patterns that you just wouldn't be able to see by looking kind of just at tissue alone, the way we more conventionally sample. And then speaking of water, so organisms in arid environments are very beholden to it. And nitrogen-fixing bacteria are no example to this. We know from kind of greenhouse manipulative experiments that they're very water sensitive. And it just so happened that I was fortunate to do this experiment over the course of the onset of a drought. So in this 3-year period, the amount of summer precipitation in this encroached ecosystem dropped by half. And we went from something that looked a little bit like this to something that looked a little more like that. And I'll explain in a second why I don't think it's necessarily valid to compare the leaves of different species to each other, but that doesn't mean that we can't look at patterns. And the non-fixing species growing in this environment maintain a really constant foliar isotope value over time. They just have the one soil source, remember. Nothing really changing there. But Prosopis, this fixing species, in years when it was really wet, has a delta value that's much closer to that of depleted fixed nitrogen. And over time, as it gets drier and drier, it increases by 4 per mil. Like that's a pretty big change in plant world. And I think also speaks to Jim's point about being careful about taking one number and applying it to a whole species. But I think this change makes sense in terms of what we might ecologically expect, that there could be water limitation controls on nitrogen fixation that then show up in this isotopic record. And another source of variation that we know again from sort of crop studies, lab studies, greenhouse studies, is genotype. So obviously plants, particularly wild plant populations, have genotypic variability, as do their bacterial fixing symbionts. And the specific genetic combinations of those can lead to really different fixation efficiencies. And again, that's something we know from the lab, we know from crops, but we haven't explored in the field, and certainly I didn't actually even mean to explore in the field until I looked at some of this data. And what's apparent is that the sort of isotopic signature of plants is really consistent over time. So if you sample the xylem sap, each of these is one plant, of a plant in 2011, then there's some seasonal up and down, you come back a year later, you come back 2 years later, it has a really consistent isotopic value. And that line, it's not explained by soil isotopes, it's not explained by plant age, it's not seen in a non-fixing plant. And what it looks like is that there is this consistency so that some plants just inherently fix more than others, potentially. They lie closer to this zero signature. And I would propose that maybe this strong interspecific variation in isotopes might be reflective of the fact that you have variable fixation within a population. Again, something interesting when you think about how to sample these things, if that's not something you'd been taking into account. And then, in order for me to sell you on the idea that sampling leaves might not be as interesting as a much more expensive, time-consuming, resource-intensive method, I was relying on the assumption that probably different plants actually don't fractionate nitrogen all that similarly, because, as I mentioned, plants differ in their turnover, in the way they move nitrogen inside their bodies, in a way that probably has isotopic implications, but that we don't test in field settings because it's challenging. And so to get a handle on whether different plants actually do have important internal fractionation, I looked at, this is big delta now, and it's the difference between the delta value of xylem sap and of foliage in the same plants at the same point in time. So this is the fractionation that happens as nitrogen moves from xylem to leaves. And in Prosopis, you can see there's a difference. It varies a little bit, but generally leaves are more depleted than xylem sap. Fair enough. But then if you look at the non-fixing species, it's like not only does it also have fractionation, but it's in completely the opposite direction. So these plants clearly process nitrogen internally in ways that strongly influence the delta signature of their leaves. And to sort of explore what the implications of that actually are, this FOLIA method that I mentioned at the beginning, it uses those isotope values to calculate this term that is the amount of nitrogen that a plant is deriving from fixation. And if I run those numbers using these two plants, usually use more reference species, but you get the idea, I'd conclude that Prosopis gets 60 to 70% of its nitrogen from fixation, depending on the time of year. But if I then take into account that the fractionation within these two species is vastly different and rerun those calculations, I'd come to a really different conclusion. And this difference in fractionation, although, as I mentioned, there aren't many numbers to go on in the field, we've done some sampling across species in northern Australia, and the general conclusion is that it can vary a lot between plants, so that leaves are not necessarily a good indicator of nitrogen that these plants have recently acquired over any time lag, even if you take into account the fact that leaves are synthesized over time. Oh, I'm running a bit late. So the bad news here is that the easy way doesn't necessarily work. That we found that assumptions that the soil signature of nitrogen that plants can access, it's not consistent over time or over space. There's some data that I didn't show showing that it can go up and down seasonally. We also have evidence that different plants fractionate nitrogen in vastly different ways, even within the same ecosystem. But the good news is that this more complex sort of sampling approach gives us more nuanced information that was obscured if you don't do things like look at this soil baseline. And although this doesn't generate absolute fixation rates, it gives us insight into, say, some of what the abiotic and biotic controls are on fixation in this common species. So we saw there's a positive effect, potentially of tree age, a negative effect of environmental processes like drought, and then also evidence for some sort of large interspecific variation that we may not have expected otherwise. So this work was mostly done with Jed Sparks at Cornell University and Tom Boughton, or this was done at the Texas A&M AgriLife Research Station down in Lacopita, Texas. And I'd also like to thank some of my collaborators in Australia who helped develop some of this isotopic method as well. So thank you. Although I do have way more graphs if you want more graphs.
Okay. Thank you, Fiona. And I'll ask our timekeeper how much time we have for questions. 10 minutes, okay. So thanks very much. Are there questions? And if there are, I'm going to hand you the magic cube so your question can be recorded. Questions, comments?
Great talk. I'm curious how you explain the soil delta N15 values as a function of the age of the plant. What you think the mechanism might be there.
Yeah, so interestingly, as I mentioned, these plants are obviously fixing a lot of nitrogen, and the bulk soil value, so if you just took a soil core, the bulk soil value goes down because they're adding in all of this isotopically light organic matter from fixation, which is one of the problems of looking, a lot of people look at bulk soil, but in some ways it's often really different to what the actual available pool of nitrogen is. So a lot of that bulk material is literally just like leaf fragments that haven't even broken down yet. But once that nitrogen is broken down, mineralized, kind of into an available sort of soluble pool, then it can be really strongly impacted by processes like gas loss in particular. So gas loss is really fractionating. It tends to preferentially take off the lightest nitrogen, and the pool that's left behind enriches more and more over time. And so I think that probably changes in nitrogen cycling that are associated with, the microclimate's different, the soil is now shaded, it's moister, it's got more nitrogen in it, it's got higher microbial biomass, that probably microbial processes like, microbial gas loss and things like that tend to move it in that more enriched direction. Yep.
Choice of colours, I don't know. Thank you, Fiona, for that. I think you've demonstrated very well that looking at the intra-individual variability can be very informative about fractionation. I wondered, did you try to do the same thing with your soil nitrogen pool? Because from what I gather, you've extracted basically the whole soil nitrogen pool. Did you attempt to characterize that in terms of mineral, organic, either in prevalence or the isotopic variability?
Yeah, so those values that I was showing, they're just for ammonium and nitrate. So there are some weird plants in some ecosystems that can access other forms of nitrogen, but generally speaking, most plants are just accessing ammonium and nitrate. And those were the concentration-weighted ammonium and nitrate values. Although some plants admittedly prefer one over the other, but I did the analysis in various ways, and it didn't really shift the conclusions.
But did you see Did you see a change in those prevalences with the age of the tree, as it is actually influencing its micro
Certainly, the concentration of that pool increased hugely, 2 or 3-fold. I think it's been a little while. That the proportion of ammonium relative to nitrate stayed relatively constant. Was that your question?
Yeah.
Yeah.
I'd be interested in that.
Yeah, the source breakdown between those two didn't change hugely. Yeah. I'll check the paper.
Another question? Yeah.
Just to make sure I didn't miss something, I was understanding what you were saying. The ontogenetic sequence, or the change within the nitrogen fixation species that you were attributing to a difference in, saying, okay, I'm trying to remember what did you attribute the difference to in the older ones?
Oh, so
the
signal looks like those older plants are acquiring from somewhere more isotopically light nitrogen.
Right.
And so I was attributing that potentially to just greater inputs of fixation.
Okay.
But you could see that same pattern because older trees, they were able to fix more, say, because they had access to, better water sources. The phosphorus is a limiter in this ecosystem as well, and older plants have access to deeper pools of soil phosphorus, things like that. Or that you could have had differential survival. So potentially the reason why old plants look like they're fixing more is because they survived, because they were a higher fixing genotype, whereas you had more mortality up to that point in plants that didn't fix as much. But either way, the general signature is that what I think is the most probable interpretation of that is that you have greater nitrogen fixation inputs in older trees.
Okay, I think you sort of answered maybe where my question is going anyway, but in terms of, there's other things, of course, with bigger trees, they're going to have deeper root systems and stuff. So
yes.
Do we know how the nitrogen varies?
Yeah,
with
and
depth?
it actually would tend to have totally the opposite effect. So nitrogen tends to enrich down the soil profile. And so if you were just looking at the fact that those roots were accessing nitrogen deeper in the profile, you would see exactly the opposite effect. So I don't think it's access to deeper nitrogen. There's not there's not a huge amount of nitrogen deposition, for example, at this site. I don't think you could attribute it to something like that. You could argue that maybe mycorrhizal colonization changes substantially over the lifetime of the plant. That's something I thought about, but I can't find any evidence for anywhere, so yeah. Yeah.
Other question?
So I was wondering about the symbiosis in the nodule itself. So it's a carbon-nitrogen trade-off, right? So they're trading carbon for nitrogen.
Yep.
And I imagine that symbiosis kind of becomes in the realm of a parasitism during low water, possibly, or?
That's a good question. So
Or maybe
there's
the
a
rates
lot
go
of,
down?
and it's actually, not that well nailed down, especially in trees. There's a lot of discussion about whether plants are obligate fixers, like they don't have a choice but to engage in the symbiosis, or whether they have some control over it. They can kind of ramp it up and down. When it's really dry, it also tends to be really hot, and I think to some extent there's just the physical bacteria aren't very active when it's very hot and very dry, and it gets dry really deep into the soil profile here as well. The question as to whether or not they sort of start to display parasitic behavior.
Or would you just see a ramping down of fixation as?
That's what it looks like, yeah. I mean, it certainly looks like that if a non-fixing plant, that if there are any sort of environmental changes in the composition of the soil pool, that they should affect both species, but in the non-fixing plants, they were very consistent over time. And the fact that the fixing plant moved towards a signature that looked more like a non-fixing plant, although, like I said, I don't want to compare them directly, but the trajectory, just looked like that there was much less fixed nitrogen entering the xylem stream. Whether that be because maybe the plants are growing less when it's dry, they don't have as much photosynthate to allocate to the nodules, whether it's physical effects of the bacteria being unhappy and ramping down their fixation, whether, also if you're not growing very much, you don't need very much nitrogen. So, if it's a facultative process, they could ramp down the carbon supply for that reason, yeah. But the physiology is a whole different, currently highly contested ballgame, so.
Do you have any cores into the trees where you can look at the wood 15 in back through time? Because there's so much variation in, say, the xylem with the age of tree. You could imagine that there are different trends that might exist for individual trees.
I really wanted to do that and was hoping it was going to be the second chapter of my PhD. Unfortunately, unlike carbon, which it's mostly in wood rings, it's fixed in cellulose and it doesn't move around. Nitrogen does move around in wood, and so it doesn't retain a faithful signature of when it was laid down. People have been working on that. It's kind of a Duncan Menge's lab at Columbia is doing some stuff with that right now, trying to see if you can get rid of all of the soluble nitrogen and just look at what's structural and left behind to see if you can use tree rings as a record that way. But those methods are not very well developed yet, unfortunately. So I thought about it, but I was thwarted.
I could concur with that. We've done labeling studies in whole forests, and the labels move radially
Yeah.
pretty dramatically, actually. And I think it's ray parenchyma cells that are moving it back and
Yeah.
forth.
So.
So it's, yeah, you'd have to look for something that was, and also water moves through multiple xylem rings, right? And the rates differ as you move distally and radially.
Yeah.
And so it's pretty complicated.
I think there's one just behind you. We might have time for one more. Oh.
Well,
All right. So this actually is not directly about what you've been doing, but you made me think about something.
Cool.
Which is when you think about the sources of variation in the soil and then what the plant is taking up, it made me wonder, have you thought about, or is anybody working on whether adding mycorrhizal nitrogen that's coming through mycorrhizae might change the signal, as well as
Absolutely.
whether you could, those mycorrhizae that are able to take up organic, whether you could get some proportional through using these sorts of approaches.
Yeah, absolutely. And people have definitely worked on that. You see different levels of fractionation with different mycorrhizal types. Like if you plot out all of the world's plants and just group them by mycorrhiza, you see their nitrogen signatures separate out. I think to some extent it's mediated not only by your mycorrhizal type, you're right, and then what kind of nitrogen is it accessing. Is it accessing organic nitrogen, which probably has a really different signature from like ammonium and nitrate. And that's one of the reasons why different mycorrhizal types do separate out differently. But then there's also, I think, probably some effect of how much mycorrhizal colonization you have. You know, is it 95% of the root system? Is it 20% of the root system? We know that different plants vary along that scale, but it's a really difficult thing to quantify, like to put a good number on, particularly when you're trying to sample something like a tree root system. So a lot of what we do know comes from more sort of pot-based studies, but certainly mycorrhizae can have really strong impacts on that. These species I was comparing were all the same mycorrhizal type, so kind of apples with apples. Yeah, great.
Well, let's take this moment to thank Fiona again. Thank you, everyone, for keeping on time. A wonderful beginning to the day. We have a 15-minute break now, and then we're going to please be back in your seats in 15 minutes, and we're going to go up a trophic level.